It is my pleasure to introduce our technical session chair person, Dr. Amita Chandan Srivedhi, an accomplished botanist, dedicated educator, and innovative researcher. Currently, she is an assistant professor at Satish Pradhan Gyan Sarna College. Dr. Amita holds PhD in botany and from the University of Mumbai, dedicated to CSIR net qualified scholarship. She has over a decade of teaching experience across several Pradhan colleges. Her work spans traditional plant sciences and modern technology. Most notably, she holds a patent for an AI-based plant leaf detection device. She is also the author and editor of several books and has published widely on focus and modern technology. Beyond academics, Dr. Amita is deeply involved in environmental initiatives and student development programs and making her well-rounded contributor has published widely on focus and modern technology. Please join me in welcoming Dr. Amita is deeply involved in environmental initiatives and student development programs. Due to time difference, our keynote speaker, Dr. Hemant Tripathi, is unable to join this live session, but he has shared of his views with us. Please join environmental initiatives and student development programs. Due to time difference, our keynote speaker, Dr. Hemant Tripathi, is unable to join this live session, but he has shared of his views with us. Please join environmental initiatives and student development programs. Due to time difference, our keynote speaker, Dr. Hemant Tripathi, is unable to join this live session, but he has
Good morning, everyone. Suprabhat.
Due to time difference, our keynote speaker, Dr. Hemant Tripathi, is unable to join this live.
My name is Hemant Tripathi. Good morning, everyone. I'm an ex-student of Thakur College. So it's a great privilege and absolute pleasure to be here and to speak with you. Sincere apologies. I'm not able to present this live to you due to some personal circumstances. It's a great privilege. Also, I'm based in the UK. Absolute pleasure. So it is pretty early for me here and to speak with you. But here I am through this recorded live presentation due to some and I'm going to talk about some of my work that is mainly around in the area of ecology, biodiversity, land use change. But here I am, and how that can relate with the theme of the conference, which is Transforming Bharat and the role of commerce science and humanities. The way I've titled this topic is Forest to Folk and the Future. So Forest to Folk is basically my work. I've last almost 20 years I've been working in the area of forestry, biodiversity, land use, and forest to folk. On the way, I've also gathered forest experience in AI and big data. So how all of that leads us to future and how commerce science and humanities converge to transform India, transform Bharat. And using all these interesting niches, how all of that, how we can solve some of these major global challenges, and how maybe biodiversity losses, food security, climate change, how we can address these challenges in India, but also across the globe. So how we can solve some of these. I can give some broad introduction of mine later on, but let me quickly jump onto the structure of this presentation in India. So in the next few minutes, I'm going to try and cover a range of things. So I'll start with what I am, what I'm doing right now. After that, I'll discuss in brief about my journey especially in last 10 years, and then what I'm doing these days, what hats I'm wearing, and what is a specific niche that I'm occupying now, and how it all relates to the idea of transforming Bharat, how we can merge or integrate the two elements of commerce science and humanities, and address some of the major challenges that I mentioned earlier in the 21st century. So in short, I think I've already covered a bit of my introduction. Right now, I'm occupying positions at the University of Leeds. I'm also a co-founder in two startups, and I'm also building a tech team for a carbon project developer as a head of tech and science. So I think we have already covered the first step. I can quickly go into the journey that I've taken over 10 years. My life in some ways feels like has evolved in two phases. So I think everything before 2013 and everything after 2013. So before 2013, I was mainly based in Mumbai, in India. And from 2001 to 2005, I was a student at the Thakur Science College. I did botany as my specialization, I remember. And pre-2013, my life was all about exploring. You can see the beautiful picture of northern Western Ghats in Pune Science College. So I used to explore lands across all these ranges. And that kind of primed me for all the interesting ecology and biodiversity work that I can see later. As you can see from these interesting pictures, it's very old across all these ranges, almost 15-20 years. I spent significant amount of time talking about nature to children, but also exploring some of the very rare flora in various parts of Western Ghats, but also coming across some interesting fauna. And that kind of built this curiosity in me about nature to children, ecological interactions, how not just species, but how biotic, abiotic, humans and non-humans interact with interesting fauna and create this term called nature that we often think that nature is devolved of humans, but actually nature is a integration of human, non-human, and all sorts of things. It's more of a sociological complex that we often think that nature is. And during this pre-2013 days, nature is this is what I get. Integration, my experience in the field, I learned very deeply that what we call nature is actually a very complex, interwoven web of life that connects everyone and everything. And 2013, I moved to Edinburgh. Very deeply got what we call interesting scholarship called Principal Development Research Scholarship that funds people to do interdisciplinary, multidisciplinary work. So I went to Edinburgh 2013 and studying interesting scholarship, Savannah College Development Research. You can see on the left is the Mukane woodland, which was the topic of my study for at least 5 to 6 years. And on the right, you can see elephants, which is the most important or most commonly found species in these woodlands. And humans and elephants interact a lot in these woodlands, and my research was all about understanding the dynamics of land use change, biodiversity, and drivers of land use change, and what it means for people, not just animals, and not just for ecosystems, but what it actually means for people in terms of their well-being, in terms of their livelihoods. Woodlands, and my research was all about understanding. And as I said earlier, humans and people and communities are part of nature, drivers of the systems that I studied, and very quickly I got inclined towards the idea of observing nature, what it actually in non-protected areas or human-modified areas where human and nature are made to coexist. They may not choose to coexist, but they are made in some way and conquest of space, and they are made to coexist in these lands. And these human-modified non-systems are basically the most commonly found systems across the globe. Like there is not all areas where animals or nature exist is protected. They may not choose to coexist. So I increasingly got really interested in space and the dynamics of these biodiversity and humans in these non-protected human-modified systems across the globe. Like there is, and my PhD resulted in some very interesting capacity building in myself. So from botanist in 2013 or pre-2013 era, I was slowly becoming a more of a you can say a landscape ecologist or a biodiversity modeler. So during my PhD, I learned a lot of interesting things. My PhD mainly revolved around understanding charcoal production, agriculture expansion, human and elephant coexistence. I was slowly on biodiversity, on ecosystem services. So if you do not know ecosystem services, are basically services that we get from different systems. For example, temperature regulation, climate regulation, human and elephant coexistence, and pollination and pest control. All of these are interesting ecosystem services that we get from nature. So my study was mainly around understanding the dynamics of these ecosystem services and how people benefit from it, and how when we because of land use change. And when I say land use change, I mean that from a more natural land cover, when we transform, for example, forest. So when we transform forest to agricultural land, or when we selectively take out trees from existing forest, what happens to animals? What happens to the forest itself? And how human communities living around these ecosystem, how do they cover? What are the impacts that they have to or when we manage. So my study revolved around this. What happens to animals? What happens to the forest itself? During my study, I used several interesting species level modeling techniques. That is where I built up some capacity on how to model species distributions, how to put multiple species together and model them together. And as you can see from this interesting chart that my study proved, there was this belief that humans and elephants both, in some ways, degrade a model because they both destroy trees, and they both might be resulting in loss of biodiversity. My study proved that the type of biodiversity that is lost is different in humans and different in elephants. And as you can see from this chart, all the red dots are species that are negatively affected by humans or elephants, and the blue dots are all the species that are positively affected by humans or elephants. So you can see all the blue dots are the species that like human elephants. For example, sparrows and a lot of small birds, passerine birds. They can survive in human environments, but all the forest moved to Leeds University, where in my PhD, I increasingly studied understanding agriculture systems. And during these times, I tried to understand how agriculture is increasingly dependent on biodiversity. It was very surprising to know that almost 30 to 40% of food production or crop yields are lost just because of crop pests and diseases. If there are natural enemies or biological control or by predators in an agricultural system, this loss in crop yields is reduced to just 10 to 15% instead of 30% or 40%. So our project here in some ways highlighted the role of biological control in not just providing these ecosystem services, supply chain or supply shock point of view. So in COVID, all the major input suppliers and even sometimes the buyers of grains or food commodities, the intensity of buying and selling or supply was reduced. So we studied how these farming systems coped to these supply shocks or risks. And we also tried to understand what is the perception of farmers in terms of their in terms of biodiversity values in their farms. And based on all of these studies, we developed a very interesting tool called iFeed tool, and the iFeed tool basically track different parameters and see projections. Like if you drag, let's say, rainfall, or you move soil parameters left or right, you will see projected change in agriculture production. So this tool was extremely useful for a lot of policymakers in deciding what policies to formulate. And then one very interesting outcome of this study was analysis of trade-off and synergies. So for example, into easy to grasp, understand, building a community around it, and there is a huge scope. And likewise, in the concept of eco-basics, where there's a need for field data collection, there have been several schemes launched by the government, for the several international organizations as well. And on the ground, impact assessment of these interventions is basically very low. And the major reason for that is that there's a lack of capacity and knowledge among especially Indian youth in going on the field and collecting data. And through these initiatives, there's a need to build that capacity. For example, we need more ecologists, we need more botanists, we need more socioeconomic data collectors who can go on the field, collect data, build applications or bundled services out of it. And this itself can be a significant innovation. So in summary, so all of these 5 points that I mentioned, they address some global problems, and when these are developed in Indian conditions, they are in ways Indian solutions to these global problems. And you can see that by focusing on the One Health aspect, we'll be able to be in a significant position to prevent any pandemics, because we'll be more prepared with One Health policy, and we'll be able to detect any potential pandemic-like situation in advance. By focusing on nature finance, we'll be able to mitigate and adapt to climate change much better. By focusing on, for example, agriculture advisories, we'll be able to improve our food and nutrition security, because our crop yield losses will be low. Our farmers will be better prepared. And again, by improving the data discovery rails, data discovery infrastructure, data infrastructure, we'll be able to be very inclusive in data and finance aspect, and that will allow us to be in a front end of AI innovation. And likewise, by promoting field data collection, expertise aspects, will be able to create a lot of green jobs and cultural stewardship. So that's the end of my slide. To conclude, so basically we can, by weaving this commerce, which is the capital, the evidence, which is science, and values that comes from humanities, into every layer of different things that we're doing. So, for example, based on my work, I could connect all of these into my layer. Likewise, other speakers or other experts who will be part of this conference would be able to connect to capital evidence and values in a more objective way, and we can use these to tackle our challenges, and these challenges in India that we tackle could become prototypes, and they could provide scalable, they could be scalable prototypes to solve all the problems at even at the planet level. So thank you very much for providing me with this opportunity again. Uh, um, you can see my email ID here. Feel free to write to me. I'll be able to respond to any questions that you might have. If you want to discuss anything, I'm happy to jump on a call and discuss. Thank you very much. Have a nice day and a very good conference.
Thank you so much, Dr. Tripathi, for your insightful views. Now, I would like to invite Dr. Amita ma'am to chair this session, and with her permission, we can start the presentations. Amita ma'am, am I audible?
Hello.
Hello. Amita ma'am, are you there?
Yes, yes. Yeah, I'm there.
Okay. Ma'am, shall we start with the presentation?
Yes, ma'am. Sure.
Thank you so much, ma'am.
Yeah, yeah. Thank you.
So our first presenter is Suhana Shetty. The title is Production of Hydrophobian BMH31 from Pseudomonas using biofilm bioreactor. You can start. Suhana, unmute yourself.
Yes, ma'am. Shall I start?
Yes, Suhana, you are audible. Yes, you can start.
So, good morning, everyone in the panel. I am Suhana Shetty, and today, along with my team member Arya Shingade, I will be presenting to you my research paper on production of hydrophobin VMH31 from the strain P. austriatus using biofilm bioreactor. So, these are the contents that I will be presenting in front of you today, starting from abstract, keywords, literature review, objective, hypothesis, methodology, recommendation, conclusion, and then we can proceed to the Q&A. So abstract, I would like to give you a small introduction sort of to our paper. So hydrophobins, production of hydrophobins is where I start from. Hydrophobins are small surface active proteins which have both hydrophobic and hydrophilic properties. Because of these properties, they help in stabilizing foams and emulsions, and a coating of these hydrophobins can also make materials more bioavailable, like a coating of these on hydrophobic drugs can make them more bioavailable, and it helps in absorbing them into our bloodstreams. So that's about hydrophobins. And how they perform is by lowering the surface tension. So they are also called surfactants. And yeah, so our study also focuses on the major challenges in the upscaling of these hydrophobin production on industrial basis. So yeah, these are the keywords that we will come across in my research article, like hydrophobin, bioreactor, pleurotus ostreatus, surfactants, foam fractionation, STS page technique, TFA, and Orbitrap high resolution liquid chromatography mass spectrometry. So to present you with a literature review of my paper, this article that I have presented to you, we have used a grass strain, a generally regarded as safe strain of an edible mushroom called Pleurotus ostreatus, and from that we have extracted a hydrophobin called VMH31 hydrophobin, since it is the most express extraction from the fermentation broth itself. So, as a result of our study, we have found that the yield is higher uh, in mycelia-based extraction, with but it gives us a lower purity as
Suhana,
compared to the.
excuse me. Please
Yes,
start
ma'am.
the slideshow first. The slides are not moving.
Uh, is the screen not visible?
The volume is little bit. Please
Uh, were the slides not visible? I have shared the screen. Hello.
Yes, sir, slide is not visible. Please share the slide.
Slide. Now,
Now it is visible.
start the slideshow.
you can see the change of slides, right?
Yes, yes.
Yes,
Please
now we
continue.
can see.
Okay. Shall I start from the beginning?
No, continue,
No,
Suhana.
you can move. Huh?
Thank you. So, from last where I stopped was the literature review, and as I've said, we found that we had a higher yield from mycelia itself, but it showed lower purity. Whereas from the fermentation broth, we have a lower yield but a higher purity rate. And the analysis technique that we used is STS page. This was used to determine the yield and the purity as well. And it was found from our research that the major issue or the difficulty that was found in production of VMH31 or any hydrophobin on a large scale was excessive foam generation. And our research article, mostly highlights the points or a solution for this problem that we faced. This was the objective of our research, to first isolate and purify the strains to compare the yields, and to mainly focus on the new proposed design for our bioreactor, for our biofilm bioreactor. From our proposed design, what our hypothesis is based on and what our research is based on is on the fact that a biofilm bioreactor enhances the VMH31 production. Why? Because it firstly forms the biofilm. It has so the strain has a layer to grow on, like it does so in the natural environment. instead of just being instead of just growing like an emulsion in a fermentation broth. Our design also aims to reduce the foam formation, which will also give higher purity as we'll be able to extract in a better quantity. This was the research methodology that we followed. We first started from the cultivation. So first, P. austriatus, as the strain I have mentioned before, was cultured in the PDB broth. So that was the first step in our methodology, followed by the extraction of VMH31, as I earlier mentioned, from both mycelia and fermentation broth. After extraction, we purified it, brought it to the most concentrated form by centrifugation and lyophilization, again from freeze drying. And then we came to the SDS page method. That technique was used to again characterize the yield. So what we finally recommend or we propose is a biofilm bioreactor design. So the design will be such as there will be a bioreactor vessel, and instead of a normal submerged fermenter that is mostly used, we will have in the headspace, we will replace the stirrers or the stirring system, the typical stirring system, with a metal structured packing. So what that will do is allow the P. austriatus strain to grow on a natural surface. As I said, instead of just being in an emulsion or in a liquid broth culture, we will also install a peristaltic pump at the bottom of the bioreactor so that the media can keep recirculating without the presence of an actual stirrer. And again, we'll also introduce air from the bottom, like instead of the head space, it will be from the liquid layer itself, with the liquid that is present below the metal structure, so that will help us in reducing the foam generation, the excessive foam generation, which was the major issue found in upscaling. And finally, to redistribute all the media, we will introduce injector tubes at the top. So, and in upscaling, we will also replace the media every 48 hours. This whole process will be repeated twice to get the best results possible. And now I come to the conclusion part. So what I want to conclude with our presentation is that we have suggested hydrofobins, as I mentioned earlier, has great industrial applications, and we have suggested a novel bioreactor, biofilm bioreactor design for the production of the same, as it helps in, first of all, biomass retention. So there is less of the washing away of the P. austriatus strain or any other strain that has been used. And secondly, it also helps in lesser foam generation. It aids in reducing the foam generation, which is the major problem faced in industrial scales. And another benefit, you can say, or a less one of the drawbacks, will be that we can also be able to use the traditional probes that we have been using earlier to measure or monitor the temperatures and the pH and all the necessary factors that are required for production of any hydrofobin itself. Thank you. And if questions, please ask.
Just small question, Suhana. Yeah, is your project, is it cost effective?
Yes. So what we have done is, instead of taking a new complete different vessel, what we have done is the earlier submerged fermenter. We have just replaced the parts and moved it from that itself, derived it from the previous submerged fermenter itself.
Okay. Thank you.
Thank you so much. Any other questions?
No, that's it from my end.
Thank you, everyone. Shall we leave the meeting, or we have to stay back?
It's up to you.
Our next presenter is Miss Sanika Save.
Sanika is there.
Ma'am, she is trying to join the meeting. Ma'am, Sanika Save is not available. So our next presenter will be Miss Ritika Gauri.
Okay, next presenter is Ritika, right?
Yes.
Yes,
Participant
ma'am.
number 3. Okay, if she is available, we can start with her presentation.
Ritika,
Yes,
you
ma'am.
can
Yes, ma'am.
present
Good morning, ma'am. Am I audible?
Yes, yes, you are audible.
Yes, 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 ma'am. Good morning, ma'am. So today's topic is research topic is investigating the role of microbial dark matter in marine and garden soil health and ecosystem resilience with the help of Dr. Uday Bhan sir and Miss Aishwarya ma'am. Introduction.
Please share
The
your.
screen is visible, ma'am.
Yeah, screen is visible, but it is moving so
Okay,
fast.
ma'am.
Yeah,
Yes, yes, ma'am.
go to the
Sure.
introduction part. Yeah.
Yes, ma'am. The microbial dark matter is uncultivable and crucial from ecosystem function present in marine and garden soil, involved in climate regulation and plant microbe interaction, still largely unexplored and uncharacterized. The literature review of marine soil, impact of climate changes in affecting of marine ecosystem through rising temperature, ocean acidification, and other stressors. Research by Francois et al. Suggests that unculturable microbes may possess unique metabolic pathways that allow them to adapt extreme conditions, thereby maintaining marine ecosystem stability. Second, role in carbon sequestration. In marine ecosystem, microbial dark matter plays an essential role in the ocean's carbon cycle, particularly in carbon sequestration. Marine microbes, including unculturable species, help break down organic carbon compound and facilitate the transfer of carbon to deeper down organic carbon compound and facilitate the transfer of carbon to deeper ocean layers where it can remain secreted for long periods. This process is a critical aspect of global carbon cycling and climate regulation. The literature review of garden soil. Organic matter degradation. In garden soils, microbial dark matter is involved in the breakdown of plant residues and other organic matter, a process crucial for maintaining soil organic carbon and nutrient cycle. Many unculturable microbes have specialized enzymes that can degrade complex organic compounds that are resistant to other microbial processes. This role in organic matter degradation ensures that nutrients are recycled back into the soil and contributes to the long-term sustainability of garden ecosystem. Second, disease suppression and soil health show that soils with greater microbial diversity, which includes microbial dark matter, have a higher rates of disease suppression. These findings suggest that understanding the harnessing the potential of unculturable microbes could provide new strategies for sustainable pest and disease management. The objectives and hypothesis. The objectives are understanding microbial functions and its role, impact of microbial dark matter on soil, garden soil, and marine soil health, comparative study of marine soil health and garden soil. Hypothesis: the presence and the activity of microbial dark matter in marine and garden soil environments are influenced by environmental factors with changes in climate, pollution, and land using, leading to shifts in the composition and functional roles of these microbial communities. Material and methodology. Methodologies are samples from various ecosystems. Techniques used are serial dilution and plating. Selective medias are used in plate counts, microscopic and curvature method, and comparative analysis. Result and discussion. MDM shows high transcriptional activity under stress. Second, positive correlation with enzymatic activity, microbial biomass, lower pathogen loads. The microbial dark matter is 30 to 40% in marine soils and 25 to 30% in garden soils. The conclusion of my research is microbial dark matter is hidden but essential component of soil health, enhance biogeochemical functions that, and ecosystem resilience, promising a venue for sustainable soil and marine management. Uh, Thank you, ma'am. That's it of my research paper. Is there any questions or any doubts you can ask, ma'am?
Yes, Ritika. Um,
Yes, ma'am.
I would like to ask you, can you share me the sites of collection? Which sites you have considered for your project?
Uh, Ma'am, coastal sites for marine soil, garden soil of my college, and
Okay, so
the
nearby
nearby.
college area you are considering?
Yes, ma'am.
Okay, okay, fine.
Okay, ma'am. Thank you so much, ma'am.
Yeah, thank you.
Me आता. Our next presenter is myself, Pradnya Ingle.
Okay, Pradnya ma'am, your number is 5, I guess, as per participant list.
Yep. But
Am I
other
correct?
solo ma'am is not available right now. So
Okay, participant number
I'm going
4
to,
group is not presenting.
yes. But they may come later, ma'am. So that's why I'm just presenting myself. Okay. Is that fine, ma'am?
Yeah, it's fine. Just your volume is little lesser. Can you
Okay.
increase your volume?
Is it audible?
Yes, you are audible.
Good afternoon, everyone. I am here to present on a sustainable approach to pollution control, biogenic nanoparticles, and microbial synergy, a green paradigm for pollutant degradation. This emerging green technology combines the nanoscience with microbiology to tackle with the environmental pollution effectively. So due to increasing pollution, these biogenic nanoparticles, which are produced by using the microbes and plants, they provide an eco-friendly solution, especially when combined with microbial processes that enhance the pollution control. For this, I am presenting the graphical abstract which shows the sustainable life cycle of polyhydroxybutyrate, that is the biodegradable polymer, and its role in the environmental bioremediation. So as you can see, this PHP, it is produced by microbes and then converted into the films for various uses. Once discarded, it degrades the soil, it degrades into soil through microbial action, and then return again to the environment as CO2 and H2O and other byproducts, which are the eco-friendly in nature. Another one, same time nanoparticles which are activated by UV light, these are employed to degrade the harmful organic solvents, pollutants like the methylene blue via photocatalysis. These nanomaterials, they generate the reactive species through electron hole separation and then attack and break down the complex molecules into non-toxic products such as CO2 and water. Research supports biogenic nanoparticles as effective agents in pollution degradation. And when these are synergized with microbes, they show their improved efficiency for bioremediation process, though real-world application and scalability which are the remaining as challenges. So for this process, it requires some improvement. So as per the literature review, the goal is to explore how BNPs are synthesized and how they work with microbes to break down pollutants and to identify challenges and future direction for the widespread use of this nanomaterial. So we propose that BNPs enhance microbial pollutant degradation through increased bioavailability, direct catalysis, and this synergy offers a more sustainable alternative for the traditional methods. The study analyzes existing research on the BNP synthesis, BNP that is biogenic nanoparticle synthesis, characterization methods for that, microbial roles and pollutant types to evaluate how this synergy performs and the improvement is needed. This green or biological synthesis of nanoparticle, it emphasizes the eco-friendly approaches. The process begins with the extraction of biological substances from either plant-based sources like seeds, flower, roots and leaves, or microbial base, that is microorganisms, including algae, bacteria and fungi. These extracts are rich in bioactive compounds which can reduce the metal ions and then they can be degraded. The biological extract is then mixed with the metal ion into the nanoparticle. After synthesis, the nanoparticles are collected, their properties are analyzed through characterization techniques like SAME and TAME, often indicated by spectral data like absorbance versus wavelength. So this method is widely used or favored for being sustainable, non-toxic and cost-effective. So biogenic nanoparticle microbe synergy, it is a promising green technology for pollution control. But further research is definitely needed and essential for optimizing the performance for the real world use and enables the implementation for bioremediation processes. These are the references. Thank you, ma'am.
Oh, yes, thank you, Pradna ma'am. I just have two questions. First one is, what do you think that major current challenges are there? And second one is, which extraction method you are using for nanoparticle extractions? Thank you.
The challenges are like, ma'am, most of the hydrocarbons which are act as the pollutants, which are get accumulated in the soil as well as in the water resources, that need to be removed, that need to be reduced from the sources, and which we are using these nanoparticles so that they can effectively remove all these pollutants by their because of their nano sizes. They can easily get absorbed over the surfaces, and these are the nanoparticles which are extracted by the green synthesis method. Green synthesis method, that is the plant-based seeds, plant leaves, and stems which are used to make the nanoparticles. Then these are further extracted. Then biochar will be formed by using the biofurnace, and then these particles further reduced to the nano size. And then these are used for the process of bioremediation along with the microbial cell. They will get immobilized with the microbial cell, and they can be implemented over the soil or water sources because these are non-toxic and which are not harmful. So we can use these nanoparticles directly with the soil as well as with water.
Okay, ma'am. Thank you.
Thank you, ma'am. Our next presenter is Miss Nafisa Khan. Nafisa, you can start.
Yes, ma'am. Hello.
Yes, Nafisa, your slide is visible.
Am I audible? Okay,
You
okay.
can start.
So, myself, Nafisa Khan, and I'm a student of MSc Microbiology from Thakur College. Today, my research topic was isolation of pesticides degrading microorganisms using a Vinogradski column. It is a sustainable approach to bioremediation. So here is the introduction of my research. The increasing use of pesticides in research in agriculture has led to significant environmental challenges. This research explores the isolation of pesticides degrading microorganisms using modified Vinogradski column. Vinogradski column prepared with pesticide contaminated soil and tailored nutrient ingredients create microenvironments conducive to the enrichment of specialized microbial communities. These columns simulate natural ecosystems and allow the selective proliferation of microorganisms that are capable of degrading common pesticides such as organophosphates and carbamates. Organophosphate pesticides, they are a class of insecticides and other pest control chemicals that contains phosphorus. Isolated strands are then evaluated for their pesticide degradation efficiency through biochemical assays and molecular identification techniques. The finding demonstrates the potential of utilizing indigenous microbial consortia for effective pesticide degradation, which offers an eco-friendly alternative to conventional remediation efforts. Now, this research underscores the importance of harnessing microbial diversity for mitigating pesticide pollution, contributing to sustainable agricultural practices and environmental conservation. So, here is the literature review of my research. Enrichment of organophosphate degrading microorganisms in HR utilizes Winogradsky column prepared with soil contaminated by organophosphate to isolate pesticide degrading microorganisms. By creating the nutrient gradients, the column allows the selective enrichment of bacteria producing enzymes like organophosphate hydrolase. Isolated strains demonstrate 70 to 90% reduction in organophosphate residues over 30 days. This study emphasizes the adaptability of native microbial population to environmental toxins and the role of Winogradsky column in bioremediation research. Biodegradation of carbamate pesticides using native biomicrobial consortia focused on isolating carbamate degrading bacteria from agricultural soils using modified Vinogradski column. The column was supplemented with urea and starch to stimulate microbial activity. Isolated microbial strains exhibited over 80% degradation of carbamate pesticides within 25 days. The study highlighted the advantage of using indigenous microorganisms in tailored conditions, achieve effective pesticide degradation and soil restoration. Sustainable bioremediation of via microbial communities employed pesticide-contaminated soil to construct vinography column, enriching pyrethroid degrading microbial population. They observed significant degradation of compounds by Bacillus and Pseudomonas species which were isolated from the column. The study demonstrated how natural ecosystem can be simulated to isolate efficient pesticide-degrading microbes, promoting environmentally friendly and cost-effective bioremediation techniques. So the objective of my research is to isolate and identify pesticide-degrading microorganisms using modified vinography column that aims to enrich microbial populations capable of degrading common pesticides such as organophosphates and carbamates. Access the efficiency of isolated strains in pesticide degradation under controlled conditions and promote a sustainable and eco-friendly approach to bioremediation for mitigating pesticide contamination in soil and water system. Hypothesis is selective enrichment modified vinography column create nutrient and environmental gradient that selectively enrich microorganisms which are capable of degrading specific pesticides. With enhanced biodegradation, indigenous microbial population adapt and express enzymes that efficiently break down pesticide into non-toxic byproducts. Eco-friendly solution utilizing native microorganisms for bioremediation that reduces the reliance on chemical or mechanical remediation methods, ensuring environmental sustainability. And it has broader applicability. The approach can be adapted to isolated and study microorganisms that are capable of degrading various classes of pesticides, including organophosphates, carbamates, and pyrethroids. Microbial diversity utilization. This method leverages the natural biodiversity of soil microorganisms to address complex environmental pollutants, enhancing the scope of bioremediation. So here is the methodology. Initially, it is sample collection, then the preparation of monography column, microbial enrichment and isolation, screening for pesticides degrading activity, characterization of microorganism, optimization of degradation condition, and application and validation. So recommendations are adoption of microbial bioremediation, integration with agricultural practice, field trials and scale up, policy awareness initiative, and interdisciplinary collaboration. Conclusion: effective microbial enrichment modified monographic column successfully enriched indigenous microorganisms that are capable of degrading pesticides. The approach offers a sustainable alternative to conventional methods for mitigating pesticide contamination. Utilization of microbial diversity. Leveraging microbial biodiversity enables efficient degradation of various pesticide residues in soil and water ecosystem. Sustainability in agriculture. The method supports sustainable agricultural practices by reducing pesticides accumulation and improving soil health. And it has future applications also. The findings open avenues for large-scale application and the development of microbial-based remediation technologies for environmental conservation. So here I end up with my presentation.
Uh, yes, I just have two questions, Nafisa.
Yes,
Are you
ma'am.
there?
Yes, ma'am.
Yeah, first thing is I want to know about you said in some things that control conditions have to be maintained. Can you elaborate on that control conditions? Which conditions are you talking about? And second one is screening of the things. Which screening methods are you using? Yeah, these two questions I want to know.
Hello, I'm audible.
Yeah, you're audible. Can you,
So, hello.
yeah, can you explain?
Ma'am, will you please repeat the question? Hello.
Yeah, my first
Hello.
reading. You are audible, Nafisa. Am I audible to you?
Yes, ma'am, you're audible now.
Yeah. I want to know about controlled conditions. You in your protocol, no? You have mentioned about control conditions have to be maintained. Which control conditions are those? And second one is
Control
screening
conditions,
method.
yes. So first question, I would like to answer that control conditions such as when we set up a Vinogradski column, using a mix of natural materials and supplements to create nutrient gradient that will mimic natural ecosystem. For that, we will use soil only from the place that is contaminated from pesticide, or means the source of soil would be obtained from pesticide contaminated site only.
Okay. And screening methods.
First, we will be using different screening methods such as spectrophotometry and thin layer chromatography.
Okay, thank you, Nafisa.
Thank you, Nafisa. Our next presenter is Dr. Uday Yadav, sir.
Good afternoon. I'm audible.
Yes, sir, you're audible.
Good afternoon, one and all present over here. Myself, Dr. Uday Yadav, Assistant Professor and in charge at Department of Botany, Thakur Sahib Narayan Degree College. My topic is exploring endophytic microbiomes of Solanum nigrum and Aristolochia indica. It is a green approach to sustainable drug discovery. About the abstract and keywords, it is for a novel drug discovery, the demand for eco-friendly and sustainable therapeutic alternatives have highlighted the potential of plant-associated microbiomes as a promising resources. Endophytic microorganisms, which are the microbes like bacteria and fungi that lives inside the tissues of the plants without causing them harm. This study explores the endophytic microbiomes of two medicinal plants, Solanum nigrum, that is a black nutset, and Aristolochia indica, that is an Indian birthwort, from Sanjay Gandhi National Park, which is in Borivali, Maharashtra. Solanum nigrum is known for its anti-cancer and anti-inflammatory effects, while Aristolochia indica exhibits antimicrobial and anti-malarial properties. Endophytes were isolated from surface sterilized root, stem, and leaf tissues using standard culturing techniques, and a total 46 morphological distinct bacterial and fungal endophytes were recovered and subjected to molecular identification via 16S rRNA and ITS sequencing, respectively. Internal transcribed spacer, that is a ITS sequence, is a spacer DNA which is situated between the small subunit ribosomal rRNA and large subunit rRNA genes in the chromosomes or the corresponding transcribed region in the polycistronic rRNA precursor transcript.
Sir,
Functional
sorry to
screening.
interrupt. Slides are not moving forward.
Slides are not. Now it is moving.
Yes, sir.
Okay. So this investigation underscores the symbiotic microbial application in drug development pipeline by adopting a green bioprospecting approaches through plant endophyte synergy. The keywords for this paper is endophyte, Solanum nigrum, that is a plant, then Aristolochia indica, Sanjay Gandhi National Park, medicinal plant, and drug discovery. About the introduction of this paper, the first thing is all about the endophytes, which is a microorganism which reside inside the plant tissue without causing them harm, and it is also known as a symbiotic association. Then medicinal plant. Again, the main reason of selection of this plant, that is a Solanum nigrum and Aristolochia indica, is like it is an unexplored therapeutic plant. They have the medicinal property, but we are not counting them as a medicinal plants, and that is what the main reason I am selecting these plants. The objective is to isolate, identify, and evaluate endophytes for antimicrobial, antioxidants, and cytotoxic properties. And the study area, it is mentioned that is the Sanjay Gandhi National Park. Why Sanjay Gandhi National Park is selected? Because it is a biodiversity hotspot with a rich flora. Fuel collection conducted with sterile techniques to avoid any kind of contaminations. Methodology: the sample collection and surface sterilization, then isolation of endophytes on PDA, potato dextrose agar, and nutrient agar media. Molecular characterization via PCR, 16S, rRNA, and ITS. Then bioassay, antimicrobial, antioxidant, and cytotoxic method, that is a MTT method I have followed over here. About the result and observation, the molecular characterization, here PCR amplifies yielded approximately 1500 base pair for bacterial 16S rRNA and 600 base pair for fungal ITS. Here the successful amplification confirmed via gel electrophoresis, and it confirms the microbial presence and it identifies it. The second part of the result is the microbial diversity and abundance. Genera identified, like which bacteria, mostly it is a Bacillus, Pseudomonas, Streptomyces, Aspergillus, and Penicillium. Solanum nigrum, predominance of Bacillus, and Azadirachta indica, it is shows the dominance of Streptomyces, and it is a Penicillium. Again, it is an antimicrobial property, these microorganisms. It indicates a functionally diverse endophytic community present inside the plants. About the antimicrobial activity analysis, it is assessed against E. coli, Saccharomyces aureus, and Candida albicans. Here, Solanum nigrum extract, it gives a strong inhibition against Saccharomyces aureus, that is a 22 mm. Azadirachta indica extract, it gives strong inhibition against E. coli, that is a 20 mm. Broad spectrum activity suggests pharmaceutical potential. Again, the broad spectrum activity means it is beneficial for the maximum microorganisms. The next point of result and discussion is antioxidants and cytotoxic potential. Here, DPPH assay gives more than 70% free radical scavenging. Azadirachta indica slightly higher antioxidant potential, and MTT assay, it is a dose-dependent cytotoxicity on HeLa and MCF7 cell line. Azadirachta indica extracts more potent, especially at 400 microgram per ml. Then is to simulated cytotoxic table. Here, HeLa and MCF7 cell line viability in percent, it is present in the this table, where Azadirachta indica extract shows the greater potency on both cell lines, especially on HeLa. About the discussion, diverse microbial communities identified in both host plant, distinct colonization patterns influenced by host phytochemistry. Bioassay confirmed antimicrobial, antioxidant, and anti-cancerous activity, and it is a promising source for novel drug discovery. Conclusion: endophytes from Solanum nigrum and Azadirachta indica possess significant bioactivity, effective against bacterial fungal pathogen, antioxidants, and anti-cancerous potentia. Green sustainable source for drug discovery, and further study required. It is for compound isolation and structural characterization. Here are some references which I have used for the conducting these experiments. Thank you. Any question?
Yes, sir. Very nice presentation and very nice topic also. Just want to know about cost effectivity. All these procedures. Am I audible, sir?
Yes, ma'am, you are audible.
Yeah. So, what all those methods we are using now, are they cost effective or costly? And further study, will it be costly or again it is cost effective?
Ma'am, it is a quite costly experiment. Like for the PCR analysis and like where we are using the microbial analysis, it is quite costly. But like our college has managed to provide the facility for the same.
For your further research. Thank you.
Thank you, ma'am.
Thank you, sir. So our next presenter is Dr. Janvi Arikar. Ma'am, you can start.
Hello. Hello.
Yes,
Am I
ma'am.
audible, ma'am? Yeah, yeah,
Yes, ma'am. You are audible.
yeah. Uh, Good morning to one and all. I'll start my presentation with the thoughts that the past holds the key to our future wellness, where tradition and nature converge to nourish the body and the soul. My topic is folk medicine for gut and urinary health and ethnopharmacology study in Dadra Nagar Haveli, which is Union Territory. Uh, we have started this study with the aims and objectives that to find out the basic plants which are explored by the tribal community in these particular area, which explore for the urinary tract infection and gastrointestinal infection, where the ancient knowledge of plant and their medicinal use meets modern scientific query. This particular research is presenting health concern, ethnopharmacology, in which the study of traditional medicine and the role of plant in human health allows us to bridge the gap between ancient wisdom and contemporary biomedical science.
Can you please start the slideshow?
Yes, yes. Is it visible?
Yeah,
Is
ma'am.
it visible? Okay, okay. Yeah, yeah. This particular research is not only uncovers the valuable ethnobotanical practices within the community of this Dadra and Nagar Haveli, but also provides the scientific lens through which we can validate the understand the pharmacological properties of the plant which are explored by this community. Excuse me. Many of the remedies found in this folk medicine have already begun to reveal their scientific merits, showing promise in the treatment of common diseases or the disorders, right? So, when we have started this particular study, we have come across with the ethnic people at the tribal communities, which already using the plants for their day-to-day activities and to cure the various diseases. So why we selected this Dadra and Nagar Haveli? Dadra and Nagar Haveli is showing the rich with that rich biodiversity, and it's having the tribal communities more than 60%. So like Dhodi, Konkani, Varli, Dubla, these are few communities which uh, located in this particular area. Dadra and Nagar Haveli is merging between Gujarat state and Maharashtra state. So most of the Gujarat state Adivasi community as well as Maharashtra state Adivasi community intermingled with this particular area. So it's a rich biodiversity, as well as this particular biodiversity is more explored by this Adivasi community also. So during our survey, we have come across with the selected plants. Total, the flora of this particular area goes around more than 800 plant species. Among that 800 plant species, we focused on the basic urinary tract infection and gastrointestinal infection plants, which are utilized or used by this Adivasi community. So we find out some plants, list out some plants and go for further analysis and study of these particular plants to give a scientific support. For the for the urinary tract infection and gastrointestinal infection, we evaluate our plant extracts for antimicrobial agar-Dix diffusion method and anti-inflammatory, that is protein denaturation method. This antimicrobial activity performed in our college lab, whereas this anti-inflammatory we have tested this in the analytical lab from outside source. So the main plants which we explored for our study is Mimosa pudica, commonly it is called as touch me not. Then Abetyllon indicum, Attibola, or this is also called as a country mellow. Then Clitoria ternata, which is called as a aparajita, commonly called as a aparajita. Melostoma, that is Indian rhododendron. Then Urena lobata, that is called as also Burl Mellow, and Coleus aromaticus, it is called as a Indian mint or Indian mint or pan ova, right? So these are 6 plants which we go for their antimicrobial as well as anti-inflammatory activity. We tested our alcoholic extract of these particular plants against cell lines of E. coli, Asorius, and Candida. And we observed that Coleus and Cliteria, these 2 particular species, that is Pan ova and Aparajita, these particular 2 species shows better results with under this antimicrobial activity. Same way, when we tested these particular plants for anti-inflammatory, we come across with the same result with Coleus as well as Cliteria ternata. So with these particular findings supports the formulations which are prepared by the tribal community to cure the urinary tract as well as gastrointestinal infections, and that particular ERK or the extract which is explored or the utilized by this common tribal community is supported by these scientific evaluations. We can use these particular findings for further polyherbal drug development. Then as these particular plants are not cultivated in the regular practices, we can conserve by saying the medicinally or most important plants. So we can make a conservation practices also, and we can go for the potential of the pharmaceutical exploration also. So these are the few references which I explored during my research work. Yes, ma'am, it's complete. Finally, I conclude my entire research work. Right, this particular study confirms the pharmacological relevance of several medicinal plants traditionally used in the tribal communities of the Dadra Nagar Haveli, which is utilized for the treating urinary tract infection and gastrointestinal infection. The antimicrobial result obtained through this agar well diffusion method demonstrate that Coleus and Clitoria, these particular 2 species give the better results as comparison. A clear concentration dependent efficacy was observed. The anti-inflammatory evaluation further supports these traditional claims. These particular findings advocate for integration of indigenous knowledge system with modern pharmacological research. The diversity of pharmacochemicals, particularly flavonoids, alkaloids, saponins, suggested surgically effects that they may prove useful for the formulating polyherbal remedies. So this is about my particular research work under Dadra Nagar Haveli. Thank you.
Thank you, ma'am.
Yeah.
It is very nice topic and really very nice research work you are carrying out.
Actually, we are continuing the formulation of these particular 2 plants also, rather going for the analytical study and isolation of heat component. We prefer for the composition or the extraction of entire plant extract and formulating them as a powder drug. So you can easily utilize this particular plant in day-to-day life also.
Yes, definitely. It's really very nice research, ma'am. And all the best to you. Yeah.
Thank you, ma'am. Thank you.
Thank you, ma'am. Our next presenter is Mrs. Sonali Joshi, ma'am. Ma'am, you can start.
Thank you, ma'am. Am I audible?
Yes, ma'am. Yes, ma'am. You are audible.
Yes. Thank you. Second, I am sharing my PPT. Okay. So good morning, everyone. My name is Sonali Rahul Joshi from ZSCTS Thakur Sham Narayan Degree College, Mumbai. Today, I will be presenting my study on antibacterial activity of lactic acid bacteria isolated from probiotic formulation, which explores the use of lactic acid bacteria as a net biopreservatives. No. Lactic acid bacteria, commonly known as the LAB, are widely recognized for producing antimicrobial compounds like organic acid, bacteriocins. In this study, I isolated lactic acid bacteria from the commercial probiotic called Darolac and evaluated its ability to inhibit harmful bacteria. The LAB-derived supernatants effectively inhibited both gram-positive and gram-negative bacteria, supporting the use of lactic acid bacteria as safe, natural preservative and antimicrobial agents. The key terms that guide this research include the lactic acid bacteria, probiotic cell-free supernatant, a natural biopreservative, microbial inhibition, and Darolac. This review of literature. Lactic acid bacteria are known for forming acid-tolerant gram-positive bacteria. They are commonly found in fermented food and within the gastrointestinal tract of human and animals. Lactic acid bacteria are known to produce several antimicrobial substances, including the bacteriocins, hydrogen peroxide, organic acid, which helps inhibit spoilage and pathogenic organisms. The main objective of my study were to isolate lactic acid bacteria from Darolac, evaluate their antimicrobial activity, compare their effect on both gram-positive and gram-negative bacteria, and assess their potential as a natural preservative. My working hypothesis was that the lab means lactic acid bacteria isolated from Darolac would produce compounds capable of inhibiting both gram-positive and gram-negative bacteria. The experiment began by culturing the Darolac in MRS broth, followed by incubation at 37 degrees Celsius. Supernatant, then we centrifuge that, then supernatants were then collected through the centrifugation and pH was adjusted. These were tested using the disc diffusion method against various test organisms. The zone of inhibition were measured to evaluate the antibacterial activity. Materials included Darolac probiotic capsule, MRS media, rotary shaker, centrifuge, incubator, bacterial strains like the Staphylococcus aureus, E. coli. Then we got the result. All the lactic acid bacteria isolated demonstrate antimicrobial activity with gram-positive bacteria showing higher sensitivity. Isolate 4 was the most effective overall. For example, isolate 3 inhibited the S. aureus with 14 millimeter zone and E. coli with a 9 millimeter zone. In conclusion, lactic acid bacteria isolated from the Darolac produced antimicrobial agents that were particularly effective against gram-positive bacteria. These agents likely include the bacteriocins, diacetyl, and hydrogen peroxide. This confirms their potential role in food safety and pharmaceuticals. So I recommend further studies for the molecular identification for the LAB strains. purification of their active compounds, testing their use in real food system, and exploring the possible synergies with other preservatives. Thank you so much.
Yes, thank you, Sonali ma'am.
Thank you, Sonali ma'am. Our next presenter is Mr. Vikas Yadav.
Good afternoon, everyone.
Yes, Vikas.
Is my skin visible?
Yes, yes, visible.
Good morning, good afternoon, everyone. My name is Vikas Yadav, and my study topic is optimization study of biodegradation of cypermethrin by using Pseudomonas aeruginosa strain by statistical design. My study guide is Bharti Wadekar ma'am. Introduction. First of all, it is important to know what is cypermethrin. Cypermethrin is a synthetic pesticide which contain alpha amino group, and pesticides are substance or mixture of substances used to control pest or insect. Despite its benefits, cypermethrin has raised several environmental concerns such as persistence for longer time, bioaccumulation, and toxicity to non-target organisms, including aquatic and beneficial insects. Biodegradation of pesticide is very essential for controlling environmental problems. Cypermethrin can also be degraded by traditional methods such as chemical oxidation and photo degradation and adsorption. Objective. My objective is to find out the degradation potential of Pseudomonas aeruginosa that can degrade cypermethrin, to identify key physicochemical parameters such as pH, temperature, inoculum size, inoculation time, cypermethrin concentration that can affect the cypermethrin degradation. Statistical design experiments such as Plankett and Berman experiment design. Plankett-Berman design are also used for optimization of biodegradation condition. To determine the interaction effect between variable influence the degradation efficacy. And hypothesis is to enhance the Pseudomonas aeruginosa strain VY to capability to degrade optimization efficacy. Biodegradation efficacy is significantly influenced by environment and nutrition factor. Interaction between parameter and has a major effect on cypermethrin degradation rate. Material and method. Then first it is important first to isolate the Pseudomonas aeruginosa strain from soil, then bacterium and biochemical characteristic of Pseudomonas for identification. Then enrichment of isolate strain of Pseudomonas strain in minimal salt media, molecular identification via 16 RNA DNA sequencing, then Plankett-Berman experiment for screening 8 factors, and to evaluate the degradation efficacy of cypermethrin using analytical method. Then this is the HPLC graph we obtain after running the degradation, in which we obtain that HPLC chromatograph reveals in progression decrease in cypermethrin concentration over time, and conferring its degradation, the retention time is 6.8 minutes. Then identification degradation intermediates from cypermethrin breakdown. The graph suggests that cypermethrin degraded by Pseudomonas aeruginosa through hydrolysis and oxidative pathway first is breakdown into benzoic acid, then phenol, then 3-hydroxybenzoic acid, then cypermethrin is finally breakdown. Then Planck-Berman design that basically it's a blaster run that it is alignment search tool for nucleotide sequencing to find out closest material in the gene bank database. This is Planck-Berman design for. This design is a statistical screening that allows for the simultaneous evaluation of multiple variable with minimum number of experiment. Helps to determine which parameter is significantly influence the degradation efficacy. Then on ANOVA result. ANOVA is basically analysis of variance. It is also extended experiment version of Planck-Berman design. Then conclusion. PBD and ANOVA validates the temperature and pH and cypermethrin concentration significantly influence degradation. HPLC confirms that Pseudomonas aeruginosa strain achieve 84.4% degradation of cypermethrin under optimized condition. GC and GM analyze identify 3-hydroxybenzoic acid as a major intermediate, confirming a hydrolysis oxidative degradative pathway. Thank you, ma'am.
Yes, thank you, Vikas.
Ma'am, is there any question?
Yes, I just want to know about how you can extract the cypermethrin.
Actually, it's already present in the soil. We collect the soil sample from where this pesticide is already used.
Okay, so screening method might be there, right?
Yeah, ma'am.
What are those? How you will screen it from the soil?
Basically, we are checking the we didn't screen cypermethrin. We check the degradation, how it degrades, how much concentration present degraded by in the soil.
Okay. Okay, Vikas. All the best. Thank you.
Thank you, ma'am.
Thank you, Vikas. Our next presenter is Mr. Raju Modi.
Pradna ma'am, can you please repeat the participant name?
Mr. Raju Modi, are you there? Can you hear me? You can start.
Your slide is visible. You can start with Raju Kumar.
Yes, ma'am. My mic is not working.
Your objective slide is visible.
Audible, ma'am? Am I audible?
Yeah, you are audible. But please start with your introduction, topic name, and then continue. Currently, your objective and hypothesis slide is there on the screen.
Good morning, everyone. My name is Raju Kumar Modi. I am final year student of microbiology. My presentation topic is.
Please share your topic name slide, please.
Synthesis and production of bioethanol from waste paper using Saccharomyces cerevisiae.
Uh,
Introduction
Rajkumar,
biotechnology for
I want to know your topic name.
introduction of bioethanol from waste paper by using Saccharomyces cerevisiae. It's my
Yeah, but
topic name.
slide is not there of topic. If you can share, please, or else it's fine.
My
It is
topic name.
bioethanol production from paper waste, right?
Yes, ma'am.
Okay, okay.
Introduction bioethanol is a biofuel derived from plant-based material has gained significant attention due to its potential to replace fossil fuels and reduce greenhouse gas emissions. Bioethanol is produced via biochemical conversion process including pretreatment, hydrolysis, fermentation, and distillation. Fermentation typically conducted with yeast such as Saccharomyces cerevisiae converts sugar into ethanol under anaerobic conditions. Objective and hypothesis: bioethanol production from paper waste presents an eco-friendly alternative to fossil fuels, contributing to waste management and sustainable energy. Pre-treatment is a crucial step that disrupts the lingocellulosic structure of paper waste, enhancing enzymatic accessibility and improving sugar release for fermentation. Materials and methods: substrate with sugar of cellulose, waste paper like newspaper, office paper, and cardboard, etc. Shred or cut into small pieces of paper. Microorganism using Saccharomyces cerevisiae used for fermentation to convert sugars into ethanol, pretreatment chemicals for hydrolysis, and dilute sulfuric acid. Method: paper waste was shredded into smaller pieces to increase the surface area and subjected to dilute acid hydrolysis using sulfuric acid (H2SO4) at 121 degrees Celsius for 30 minutes. Fermentation was conducted by inoculating Saccharomyces cerevisiae into the hydrolysate at initial cell concentration of 10 into the power 6 cells per ml. The culture was incubated at 30 degrees Celsius for 72 hours under anaerobic conditions to promote ethanol production. Here is the graph of production of 72 hours fermentation. The ethanol concentration increased over time. Second graph is sugar consumption and ethanol production. If fermentation is high, then sugar concentration low. Conclusion: the results confirm that enzymatic hydrolysis of paper waste is an effective strategy for obtaining fermentable sugars with high conversion efficiency and minimal inhibitory concentration. Inhibitory effects. The ethanol yield of 22.5 gram per liter was comparable to previous studies demonstrating the feasibility of bioethanol production from lingocellulosic waste. The final ethanol product is industrial grade bioethanol strain, making it suitable for fuel and antibacterial applications. The result supports its practical application in healthcare, sanitation, and industrial settings, where microbial control is essential. Thank you.
Okay, you're done with the presentation, Rajakumar.
Yes, ma'am.
Okay, okay. Thank you.
Our next presenter is Mr. Navneet Bhagat.
Yes, ma'am. Please.
You can start, Navneet.
Hello.
Yes, yes.
Ma'am, my PPT is visible.
Yeah, but keep it in on a slideshow mode. It is bit smaller.
Okay, wait, ma'am.
Can you zoom if possible? Or else you can change the orientation. I guess you are presenting through the cell phone. Are you
Yeah,
presenting
yes,
through
ma'am.
mobile? So you can change the
Yes.
orientation. No? Yeah, landscape.
Ma'am, actually, I have opened in PDF and it's.
Acha. So can you zoom little bit?
Wait a minute, wait a minute.
Hello. Good afternoon.
Ma'am, now it is.
Yes, yes. Now it is
Okay.
better. Yeah.
Yeah. Good morning, respected teacher and my dear friends. My topic is
Your
optimization
name
and
is
fees.
your name is Navneet, right?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, okay.
Good morning, respected teacher and my dear friends. My name is Dr. Bhatti Wadekar, ma'am, and from Department of Microbiology. And today I am going to present on the topic of optimization and physicochemical analysis of citric acid production from cornstarch by Aspergillus niger NB19. Introduction. Citric acid, which is a white powder crystalline form, which mainly used in food, pharma, and chemical industries. Microbial fermentation is preferred for large-scale citric acid production. Aspergillus niger is the most efficient microorganism renewable carbon source for fermentation. Yield depends on factor like pH, temperature, nutrients, and optimizing fermentation boosts both yield and industrial feasibility. Objective and hypothesis. Objective to optimize fermentation parameters using Aspergillus niger NB19. Object to evaluate physicochemical properties of produced citric acid to identify the optimum incubation period for maximum citric acid production, to compare citric acid yield across different psychological conditions like pH, temperature, inoculum size, etc. 4 degrees Celsius, that is Pseudomonas differentials agar. Optimization of pH, temperature, incubation time, inoculum size, and constant concentration. Tested various carbons, example glucose, sucrose, analysis by FTIR, HPLC, and SEM. All experiment in triplet considers statistically significant. Result and discussion, like here is a pH effect of pH on citric acid
You're not audible.
productions, like pH is 3 yield the highest citric acid productions. Second graph, like effect of temperature of citric acid productions. A 36 hours incubation produce maximum citric acid followed by decline of citric acid yield too low, high resultant in suboptimal output. 100% cornstarch was optimal, higher concentration caused substrate inhibitions. Glucose and sucrose were the most effective carbon source. Complex carbs gave lower yields. Peptone and yeast extract were best nitrogen sources. Nitrogen inorganic sources led to the poor productions. Like this is the graph, like effect of cornstarch concentrations on citric acid productions, that how that first at 60% the concentration was means increased, and after that it declined at 120.
You're not audible, Navneet.
Different nitrogen sources. Yes, ma'am. And citric acid, this is SPLC chromatogram. And now this is the conclusion. Optimal condition pH 3 degree, 30 degree Celsius, and that enhanced citric acid yield, finding aligned with previous research confirming aspergillus niger efficiency. Thank you so much.
Yes, thank you.
Our next presenter is Ms. Krupali Varia. Muskan, are you ready for presentation?
Hello, ma'am. Yes, I am.
Okay. Amita ma'am, can we start with Muskan? Krupali is
Yeah,
not.
no problem. Actually, Muskan number is 14, right? So
Yes,
in
ma'am.
between, Baria is there, no? 13 number.
She has
She's not.
not joined.
Yeah, she didn't join yet. So we,
So, can I?
yes, Muskan, you can start.
Okay. So, a very good afternoon, everybody. I am Muskan Mansoori from Thakur Sham Narayan Degree College, University of Mumbai. Today, I'll be presenting my research study titled Revolutionizing Soil Health, Enhanced Microbial Bioremediation of BTX Contaminants. Okay, BTEX stands for benzene, toluene, ethyl benzene, and xylene. These are toxic, cancer-causing chemicals commonly found in soil, contaminated by petroleum leaks and industrial waste. They are dangerous because they dissolve easily in water and move quickly through water and soil, contaminating the groundwater and harming the ecosystem. There are very conventional cleanup methods like digging out soil and using chemical treatments, but they are very expensive and they often cause harm to the environment. So my research explores a more natural, more sustainable solution using bacteria that are already present in the contaminated soil to break down these pollutants. Here are a few keywords that are redundantly used in this paper of mine. Literature review highlights the carcinogenic nature of BTX compound as described by Singh in 2019, emphasizing their environmental and health risks. Atlas in 1984 discussed the broader impact of oil pollution in marine environments. Research in 2010 explores how compost can be effectively used in remediating the hydrocarbons contaminated in the soil. The objectives and the main goal of the studies were to identify bacteria that are already living in the polluted soil that can naturally break down the BTX compound. Next was to see if adding these bacteria back into the contaminated soil actually speeds up the cleaning process, meaning they act as a catalyst or not. And third was to find eco-friendly strategies for cleaning the polluted soil without harming the environment. hypothesis. As a researcher, I believe that by inoculating or adding communities of BTX degrading bacteria into the polluted soil could increase the speed and efficiency of the breakdown processes, leading to a significantly cleaner soil in a shorter period of time without using any harmful chemicals that can somehow just disrupt the natural flora of the soil. The research methodology, soil samples were taken from a petrol station area known for the hydrocarbon contamination. The soil was sterilized to eliminate the native microbes and they artificially spiked with BTX compound. So we have set up two experimental groups. In one, we had a control group with where there is no bacteria added, and next was a test group where bacteria known to degrade BTX were added. The experiment ran for 30 days at stable temperature and humidity levels. The bacterial growth and BTX level were monitored throughout the period of 30 days. Next was monitoring the microbial activity to understand how well the bacteria was working or not. We have measured its optical density, the viable count, and the active degradation ability. For the optical density, this which shows if bacterial biomass is present, we have observed a 30% increase, suggesting a strong bacterial growth. In viable count, the count how many living bacteria were living in the soil or not, the increase confirmed that the added bacteria survive and multiply. This result shows that the bacteria were not just surviving but actually actively helping to degrade these BTX compounds. And after active degradation, after 30 days, the control group, which had no bacteria initially, only removed about 2 to 5% of the BTX compound, but the inoculated group, the second setup, removed 35 to 45% of the pollutants, and which is like 8 to 10 times of the improvement. This shows that adding the right bacteria can make a huge difference in how quickly and effectively a soil pollution is cleaned up, which is polluted due to BTX compound in the soil. And the conclusion basically states that the bioaugmentation, which is adding healthful, naturally adapted bacteria, is a powerful and environmentally friendly solution. It can significantly reduce pollution without disrupting the ecosystem or costing too much. Because the bacteria are from the same environment, they easily blend in and do not introduce any foreign risk to the soil. This method could be applied on a larger scale to help the cleaning real world contamination, site safety, and sustainability, and also reducing the cost of cleaning up these sites. Um, Thank you for your time and attention. I'd be happy to answer any questions or discussion further. Thank you.
There is a background noise, Muskan.
Not from my
Are you done?
Yes, ma'am, I am done. Even I can hear the background noise.
I can't hear you anything. Someone else disturbance is there?
Yes, ma'am, it's somebody else.
Okay, fine. Muskan, I just wanted to ask you one question regarding
Yes,
viable count. Is there any recommendation that we can get a more viable count?
Ma'am, we can use various enhancing microbial gene enhancing techniques, which can increase the number of viable count. We can provide even more media, which will support the growth of specific microbes, which are easily degrading the BTX compounds from the soil.
Okay. Thank you.
Thank you so much.
Thank you, Ms. Khan. Our next presenter is Mr. Siddesh Pathak.
Hello, ma'am. Am I audible? Hello.
Yes, Siddesh, you are audible.
Okay, so I am Siddesh Pathak and my title for paper presentation is Recent Approaches Towards Various Copper Remedies. So first we'll see the introduction. So this research was mainly done in the context of nosocomial infection, which is the infection which is acquired through hospital visits. And as we all know that nosocomial infections are a serious global health issue, microbial contamination on hospital surfaces is a major cause of infection spread. There is a growing need for alternative antimicrobial strategies, especially due to antimicrobial resistance. And here comes our copper. So copper has a natural ability to contact kill the bacteria which are present in the environment. So we will be testing those copper atoms which are useful in inhibiting the growth of microbes. So the review of literature, so antimicrobial history. So copper has been used since ancient times for its preservative and healing properties. Clinical impact, hospitals using copper alloys on higher touch surfaces report significant reduction in infection rates. And mechanism of action, copper disrupts microbial membrane, produces oxidative stress, produces the superoxide species of oxygen and damages genetic material and leading to cell death.
हे चालू आहे तेवढी माणसं आहेत.
To check the efficacy of copper and copper alloy by testing it against E. coli culture and to confirm the bacteriostatic ability of copper and its alloy. Alloy means the mixture of metal. The research methodology. So what we do is we take two petri plates and inoculate, that means treat with the E. coli culture, and then we add one disc dipped in copper in one petri plate and one disc without copper in another petri plate. So what happens? What we observe is that the plant growth of E. coli were cultured and screened for growth of on nutrient agar media. So these two petri plates have the nutrient media in it. So one plate which was without copper disc shows heavy growth, but with copper disc shows no growth. So after that, we can conclude, after observing these results, that after performing this experiment, it can be concluded and confirmed the bactericidal property of copper and its alloy. Hence, copper material can be used specifically in hospital in order to avoid the nosocomial infection. Here are the references, and thank you.
Okay, thank you, Siddesh.
Okay. Okay, ma'am. So, do you have any questions?
No, no, Siddesh. Thank you.
Okay.
Thank you, Siddesh sir. Our next presenter is Ms. Sheetal Maurya. You can start, Sheetal.
Yes, ma'am. Good afternoon, everyone. My name is Sheetal Kailash Maurya, and I am a student of MSc Microbiology at Thakur Shyamnarayan Degree College, Mumbai University. Today, I'm excited to present my research title, Antimicrobial Potential of Mangroves, a phytochemical exploration of nature's defense against multi-drug resistant pathogens. This work is guided by Mrs. Pradnya Abhishek Igre. I will walk through I will walk you through the key findings and implications of this research, which is focuses on exploring natural antimicrobial agents from the mangroves to address plant pathogens and promote sustainable agricultural practices. Mangroves, mangrove ecosystem particularly are very rich in medicinal potential. I use the mangroves of Gorai Beach, Borivali, which is in Mumbai near Charkop. In this study, I focused on Avicennia marina, a common Indian mangrove, to evaluate its antimicrobial properties. Leaf samples were collected during the winter season and processed using oxalate extraction with organic solvents like ethyl acetate and methanol. I tested these extracts against authenticated strains that are known threats to plants like mango and cashew. Our results indicated that the extracts exhibited strong antimicrobial activities. This highlights the Avicennia marina as a promising natural alternative to synthetic chemical pesticides, supporting both sustainable agriculture and environmental protection. Previous studies have laid the groundwork for exploring mangrove species as an antimicrobial agent. Like in 1999, there was a study detailed the medicinal and economic uses of mangroves in traditional system. Later in 2009, the study demonstrated the antimicrobial efficacy of Avicennia marina, especially against agricultural pathogens. In the same year, another study explored how mangrove plants could exhibit resistant bacterial strains. These findings strongly support my current investigation and validate the selection of Avicennia marina for detailed phytochemical and antimicrobial testing. This study had four key objectives. First, to collect and correctly identify Avicennia marina from Gorai Beach region of Charco, Mumbai, Maharashtra. Second, to extract bioactive compound using oxalate extraction with two solvents, ethyl acetate and methanol. Third, to test these extracts against authenticated plant pathogens like Xanthomonas coprestiris and Staphylococcus xylosis. Finally, the data was analyzed to evaluate the antimicrobial potential and its implication for sustainable biopesticide development. My hypothesis was that Avicennia marina leaf extracts would demonstrate significant antimicrobial activities against common crop pathogens. If proven, this would support their use as natural, sustainable substitute for the synthetic chemical pesticides in agriculture. To begin, leaf samples of Avicennia marina were collected from Mangrove Beach, Gorai Beach area. Samples were shade dried, powdered, and subjected to oxalate extraction using methanol and ethyl acetate. For the microbial testing phase, authenticated strains of Xanthomonas compestris, Staphylococcus xylosus, Bacillus borophilus, and Aspergillus niger were obtained from certified cultural banks. Antimicrobial testing involved well diffusion assay to measure zones of inhibition and MIC test to determine the most effective concentration. Additionally, phytochemical analysis was conducted to detect secondary metabolites like alkaloids, flavonoids, or saponins. GCMS was also used to identify specific bioactive compounds in the methanoic extract. Here is a quick visual overview of our methodology starting from sample collection at Gorai Beach, followed by oxalate extraction, testing against pathogens, and analysis of active compound using GCMS and phytochemical screening. The results clearly showed that the methanolic extracts of Avicennia marina had the strongest antimicrobial effects, especially at the concentration of 100 mg per ml. Among the tested pathogens, Bacillus borophilus was most susceptible, showing inhibition zone of up to 22 mm. MIC tests were confirmed to extract potency with value as low as 1.56 mg per ml in ethyl acetate extracts against Xanthomonas comprestris. These effects were comparable with standard agents like streptocycline and bethane, despite the plant extracts being crude. GCMS and phytochemical analysis also confirmed the presence of important bioactives like flavonoids, saponins, and alkaloids, supporting the therapeutic potential of these extracts. Based on these results, I strongly recommend integrating Avicennia marina extracts into agricultural pest management systems. These plant-based alternatives can reduce the ecological and health impact associated with chemical pesticides. Moreover, future studies should aim to isolate and characterize the specific active compounds responsible for antimicrobial effects. Lastly, conserving mangrove habitats like Gorai Beach is vital not just for environmental reasons but also to preserve these unique bioresources for future scientific breakthroughs. In conclusion, the antimicrobial activity of Avicennia marina leaf extracts was found to be significant, particularly against Xanthomonas compestris and Staphylococcus xylosis. Both ethyl acetate and methanol extracts showed promising results. Notably, the methanol extract demonstrated the strongest antimicrobial activity comparable to chemical agents such as tetracycline and bethane. GCMS analysis identified several bioactive compounds responsible for the same activity. These findings were supported by both well diffusion assay and MIC testing, highlighting the potential of Avicennia marina as sustainable biocontrol agent for agricultural applications. Thank you so much. These are the references I have used. These are the some references I have used for the purpose. I'm now open for any questions you might have. Thank you so much.
Yes, Sheetal, very nice presentation.
Yes,
I just have
thank
two
you so much,
questions
ma'am.
for you. First one is, can you um, suggest anything to conserve the mangroves? And second thing is, how you will come over the limitations of your research.
Ma'am, for the suggestion to conserve mangrove, first of all, we cannot grow mangrove anywhere. We have to plant them in coastal regions. So the nowadays, like BMC is cutting down mangroves to do the uh, development things. So we have to, in a governmental level or BMC level, we have to stop cutting mangroves on a first place. And instead of planting decorative things around the coastal regions, we should just go for mangroves and plant them only. So it will be like mangroves don't not only protect us from bacteria or whatever microorganisms we are supposed to use them for in our research, but also they protect us from natural disasters like waves and everything. So we can, in on governmental level, we can just leave the coastal areas by themselves, and mangroves will find their ways to grow again on the coastal areas if they are extinct from there, and they actually need those environments. So we can also clean and keep the environment for their growth or then more growth of them. And what was the next question, ma'am? Ma'am, your mic is off.
Ma'am, you are
Yeah, my second question was how you will come over the limitations of your project.
Um, limitation that I faced actually was to I was not able to take the mangroves. The thing is, wait a second. Okay, the thing is, I was not able to take I cannot take the whole part like of the plant. I have to use the leaves only. The reason why I use leaves only because in that way I will not destroy the whole plant. I will just take the leaves and the whole mangrove plant will by its
But that would be difficult, no? Whole mangrove plant.
yeah, but some other studies have showed that they have taken stems also. They have taken roots also. We cannot do that from using mangroves. So we have to focus on leaves only, so that they will be
So you are
in a
focusing
way
on
they are not destroying.
pneumatophores. Pneumatophores.
Yes.
Okay. Yeah. Thank you so much, Sheetal.
Thank you so much, ma'am.
okay. So the next presenter is Sanika sir.
Pradna ma'am, is there anyone left?
Yeah, ma'am. I'm Siddesh Patel. Ma'am, Sanika is left and one Ashwarya Nagesh.
I guess. How many presentations are remained?
Ma'am, two only.
Two only. Okay.
Yeah,
So are they going to present now?
yeah, ma'am.
Okay.
Sanika, are you there in the meeting? Please respond.
Yes, yes, yes,
Sanika is
yes.
going to present now.
Yeah, ma'am.
Okay.
Sanika and then Ashwarya.
Sanika and are they from same group? Sanika is participant number 2, right? Sanika
Yeah,
Aishwarya.
ma'am.
Yes, ma'am.
Okay.
Can I start, ma'am?
Yes, yes, sure. You can start.
Okay, ma'am. Good afternoon, ma'am. My topic name is
Your slide is
phytochemical
not
screening and
properly visible, Sanika.
huh,
Only half
एक minute.
side is visible. Entire slides are not.
Sanika, zoom out the
Okay.
slides. Don't zoom in too much.
Is it now visible, ma'am?
Only half side is visible. I guess you have zoomed it. Can you just a little bit lesser the zoom?
Actually, I have clicked on a show slide.
Okay. So orientation is on landscape mode or portrait mode. Can anyone else share her slide? Saida cordifolia. That much only I can see.
Okay, Sanika, do one thing. Just wait a bit. Ashwara ma'am will present because she is ready, and you present afterwards. Okay? Is it fine with you, Sanika?
Okay.
Yeah. So the next presenter is Ashwara ma'am.
Good afternoon, ma'am. Am I audible?
Yes.
One second, please. Is my slide visible, ma'am?
Yes.
Good afternoon, everyone. My name is Ms. Aishwarya Ajay, Assistant Professor of the Microbiology Department here at Thakur Shamnaran Degree College. My topic for today is antimicrobial activity of vanadium and copper oxide nanoparticles, where I am showing a comparative study. Okay. So, an introduction. So we are talking about the rise of threat of antimicrobial resistance, which has accelerated, and over here we are trying to search for novel, cost-effective antimicrobial agents. So I have selected certain metal oxide nanoparticles, such as vanadium oxide and copper oxide, which have emerged as promising candidates due to their broad spectrum activity and high surface reactivity and the potential to disrupt microbial membranes. So this study evaluates the comparative antimicrobial efficacy of vanadium oxide and copper oxide nanoparticles, where I've tested it against 8 clinically relevant microorganisms, which includes gram-positive and gram-negative species. The aim over here is to identify the potential alternatives or enhancers to conventional antibiotics with practical relevance in clinical, environmental, and industrial application. So the nanoparticles of interest are vanadium oxide, which is a reactive oxygen species generator or process redox activity, and copper oxide nanoparticles, which is known for its membrane disruption activity and copper ion release. These two are broad spectrum and they have high potential against and compare the antimicrobial efficacy of vanadium oxide and copper oxide particles and analyzing the effect on the pigment production of Serratia macerans gram-negative organism. So the organisms used for gram-positive nature were Ms. aureus, Nocardia, Bacillus cereus, and Candida species. Even though Candida is a fungi, I have kept them as a gram-positive organism as due to its gram nature stability. And for gram-negative organisms, we are looking into E. coli, Shigella, S. typhi, and Serratia marcescens. So to test the efficacy, I have done the pitch plate method where we have prepared the nutrient agar and which was sterilized, and we have done the ditch plate method as a qualitative assessment. There I have made separate plates for gram-positive and gram-negative organisms. So the observations seen are as follows. For all the 8 organisms, E. coli, Shigella, S. typhi, and Serratia, which is your gram-negative organisms, they showed reduced growth. E. coli showed reduced growth over the ditch of vanadium oxide nanoparticles. That is, copper oxide nanoparticle did not show much antimicrobial effect. Shigella species, no inhibition and no inhibition of copper oxide nanoparticle. S. typhi, again, no inhibitions in both types of nanoparticles. Serratia macaroons had no pigment over the ditch, but pigment outside the ditch was seen. For copper oxide nanoparticles, Serratia had pigment present over and outside the ditch. For the other 4 organisms, S. aureus, Nocardia, Bacillus cereus, and Candida, we can see the observations where S. aureus, very little growth over the ditch, same for copper oxide nanoparticles. Nocardia showed almost no growth on both nanoparticles, and Bacillus cereus, no growth over the ditch for both the nanoparticles. Candida species showed growth over the ditch. So, what are the basic interpretations? Vanadium oxide exhibited a stronger antimicrobial effect, particularly against gram-positive organisms such as Bacillus cereus, Nocardia, and S. aureus, showing complete or near complete inhibition over the ditch area. Copper oxide nanoparticles showed comparatively lower antimicrobial activity, with most gram-negative organisms displaying growth and only partial inhibition, which is noted in gram-positive strains. Candida species, despite being a fungus, showed no inhibition on either nanoparticles, suggesting that these metal oxide may have a limited antifungal activity under current conditions or the concentration used. I forgot to mention one part, that the concentrations which was used for this study was ranging from 0.01 to 0.1. Okay. So Serratia macerans, coming back to the interpretation, that Serratia failed to produce the red pigment, that is prodigiosin, in the vanadium oxide treated ditch, indicating a potential metabolic suppression of or a stress response which is induced due to oxidative mechanisms due to this nanoparticle, or it can be stated as an indirect marker for the nanoparticle toxicity. So this method proved effective for visualizing the zone-specific inhibition and pigmentation changes, which was serving as a qualitative and an insightful screening tool. Conclusion. So vanadium oxide nanoparticles demonstrated super antimicrobial efficacy against gram-positive bacteria, indicating its potential as a nano-based antimicrobial agent. Copper oxide nanoparticles showed limited activity, suggesting the need for optimization of concentration, particle size, or combination with other antimicrobial agents or enhanced performance. The suppression of prodigiosin production in Serratia under vanadium oxide exposure suggested possible interference with secondary metabolic pathway, warranting further molecular investigation. And this ditch plate method was validated as a simple, cost-effective, and a visual representative assay for preliminary antimicrobial screening of these nanoparticles. So the future research where we focus on will be on quantitative assays, mechanistic studies of ROS quantification, membrane integrity tests, and cytotoxicity evaluation, which can be done either by using a CAM assay as an example. So in future work, we can use these nanoparticles as surface coating in hospitals, surgical tools, or biomedical devices to prevent nosocomial infections. And given the selection of inhibition of skin pathogens, vanadium oxide nanoparticles may be formulated into wound dressings, creams, or antiseptic gels for treating any kind of minor infections or burns. The suppression of the dye of Serratia, it can suggest potentially suggests as a potential for nanoparticles to modulate microbial metabolism, opening pathways for studying the non-lethal microbial control strategies that will reduce the pathogenicity without inducing resistance. So given the simplicity and scalability, this research could contribute to affordable nano-based disinfectants, and it can be used as a treatment aid in rural and unreserved areas, which can help us in bridging the gap in infection control. These are the references, certain references. Thank you. Any doubts?
Yeah, no questions, ma'am, as such. Just one suggestion, your font size should be a little bit visible.
Oh, okay.
Font size was not that
Visible?
readable. That
Okay.
is the only suggestion, ma'am.
Okay, ma'am. Sorry for the inconvenience caused.
Thank you.
Okay. Thank you, ma'am.
Okay, so our last presenter is Sanika Savya. Sanika, are you ready?
Yes,
Have
sir.
have you mailed or emailed?
Yes, I have mailed.
Sir, please
And
tell me the group number of
okay, ma'am.
Sanika.
Sanika is
Is it
from
2?
group number 2.
Okay.
Yeah. Sanika, have you mailed the PPT?
Yes, sir.
Okay, okay. Sanika, I can't see your mail in my inbox.
Okay.
Wait a minute.
Just a second, sir.
Sanika, I have sent you the email ID.
Huh.
Send your PPT on that email. Okay? Fast.
Okay, sir.
Thank you. Sanika, please, can you share the slide?
Yes, sir. Is it visible, ma'am?
Yeah, it is visible, but same. Half part is only visible.
Okay. I have mailed in my PPT again.
I couldn't find your PPT in my inbox. Where is it? I can't find it.
एक मिनिट तर मी एकदा परत चेक करते. सर
Yes, Sanika
Sanika,
is going to present.
have you shared the PPT again? Because I cannot see the PPT
Yes, sir.
here in my inbox. Okay, now it's visible. Okay, done. Done. 1 minute, just 1 minute.
Okay.
संपेल.
Hello.
Hello.
Yeah, now you can see the slide.
हाँ.
Okay, start the presentation.
Hello, good afternoon, everyone. My name is Sanika Save. I'm presenting a research topic on phytochemical screening and antimicrobial activity of Sida cordifolia plant. Sida cordifolia is often referred as a bala in Ayurvedic practices. It is a shrub that belongs to a Malvaceae family. This species is indigenous to tropical and subtropical areas, including regions of India, Sri Lanka, and parts of Africa. The plant is particularly used for its benefits in addressing inflammatory ailments like fever, respiratory tissue, and some disorders in nervous system. In recent years, it is used for its phytochemical screening, the investigation of antimicrobial properties as researchers and and exploit the bioactive compounds found in traditional medicinal flora. The medicinal plants represents a vast source of bioactive compounds, including alkaloids, flavonoids, tannins, saponins, and terpenoids. A methodology logic contains the collection and preparation of plant materials, where the fresh leaves, stems, and roots of Sida cordifolia will be collected from the previous distinct locations. Proper identification and authentication will be performed, and after that, the procedure will continue with cleaning and drying procedure where the clean plant will be shade dried at room temperature or air dried in a well ventilated place. After it, the grinding procedure where the powder plant material will be ground to a fine powder using a mechanical grinder or motor and pistol. The extraction process where gum extraction is continue with the use of methanol and water, where the cool dry extraction process is used. Next one is phytochemical screening, where a phytochemical screening will be carried out using standard chemical tests to detect the presence of various classes of compounds. First one is alkaloids, where a Dagendorf reagent and Mayer reagent is used. A Dagendorf reagent where the chemical test detects the presence of alkaloid in plant extract. The test involves a reagent where a solution of potassium bismuth iodide to extract the alkaloid is present. The color change color change indicates orange or a red orangey precipitate color change indicates the presence of alkaloids. In a Mayer, while using a Mayer reagent, the reagent used is potassium mercuric iodide, where if the alkaloid are present, the white precipitate will be formed. That precipitate indicates the positive test result. Second one is flavonoid, where the Shinoda test and lead acetate test were majorly performed. A Shinoda test it involves a reaction between extract with magnesium and hydrochloric acid, where the color changes to pink or red color indicates the presence of flavonoid. In lead acetate test,
Shanika, your
the
5 minutes
plant
are over.
extract.
Your 5 minutes are over.
Okay.
Thank you.
Okay.
Okay, ma'am. Amita, ma'am. Hello.
Hello.
Amita, ma'am. Hello.
Hello.
Yeah, ma'am. So the session is over now. So would you like to express your thoughts on today's presentation?
You're not audible, sir. Your voice is not clear.
Okay. Yeah, ma'am. Now you can hear my voice. Hello.
Yeah, but voice is not that clear.
Okay, ma'am. Like, are you able to hear my voice now?
Yes.
Yeah, ma'am. So would you like to express some of your thoughts on the presentations?
Yeah.
Yeah, ma'am.
So first of all the topics were really very nice, and research is there is scope to what we can say. Um, Firstly, I would like to extend heartfelt congratulations to all the participants. It's truly inspiring. All the topics were intellectual, curiosity, confidence, and dedication. Each of you has taken important steps toward becoming thoughtful researchers and effective communicators. Yeah, just the thing is that structuring and clarity should be there. Yeah, and some presentation skills I would like to suggest. And lastly, some students have given Q&A also very nicely. They were prepared. And keep refining your work. Stay curious. Continue participating in such a platforms. And yeah, you are on right path. Keep shining.
Okay, ma'am. Okay, thank you for your valuable insights. So now, as we come to the end of this wonderful and insightful conference, it is my pleasure to extend a heartfelt vote of thanks on the behalf of Jagru Singh Charitable Trust and Thakur Shamnayan Degree College. First of all, a big thank you to the speaker, to the keynote speaker, Dr. Hemant Tripathi sir, for sharing your knowledge and insightful thoughts. And thank you to the Dr. Amita ma'am. Thank you, ma'am, for judging this session. Without you, this session wouldn't have been possible. And thank you to our principal, sir, management, and organizing team. Thank you for your support and for making sure everything as we planned and smoothly managed. To all the participants and attendees, thank you for being here, for engaging with the session and making this event lively and meaningful. Let's carry forward the ideas and energy from today as we continue to learn, grow, and contribute, and making this event lively and to a better future for Bharat. Thank you, everyone. Now you can leave the meeting.
Thank you so much, sir.
Thank you.
So can I disconnect?
Yeah, ma'am. Please share the judging sheet.